Welcome everyone. Dave Ritchie here. I'm the director of the Maddie Riewoldt's Vision Centre for Research Excellence in Bone Marrow Syndromes, bone marrow biology. And if you're looking for the CRE Expert Webinar on a Monday afternoon, you're in the right place. And we've got a really fantastic speaker coming up, and I'll get on to Elizabeth shortly. So this is our fourth session in this series, and they've really been fantastically well received, and we're hoping to be able to sustain them on an ongoing basis. And we've had a range of expert speakers from around the world and locally to both entertain and educate us, and that will definitely be the case today as well. It's always polite to say thank you to the custodians or the owners or the curators of the places that we're meeting, and so that's no different for today. So wherever you're joining us from, spare a moment just to say your own thanks to the traditional owners on which we meet, which is the Wurundjeri people here in Melbourne. And we're very grateful for the opportunity to live in such a wonderful place and undertake our clinical and research activities, and we pay our respects to those people, their elders, and any Aboriginal Torres Strait Islander people who may be joining us today. We particularly thank you for your participation. So as I mentioned, this is the fourth in a series of expert webinars, and really another string to the bow of MRV. MRV has been in existence for 10 years now, and we've achieved many great things, albeit with the generosity of donors and families and the patients indeed. The generosity of the Riewoldt family and the series of boards that have guided MRV through the last decade, but also particularly acknowledging the incredible work and generosity of spirit that work has been undertaken by the researchers who have been able to undertake their work with funds raised by MRV and curated by its Scientific Advisory Committee and indeed the Executive Administration Board of MRV. And all of that big long list of people that I've just mentioned, and all of those integrated efforts and all of that generosity of spirit and activity are a recognition of a particular person, and that indeed is Maddie Riewoldt herself. And today, just by sheer coincidence, actually represents the anniversary of Maddie's death. She died today, all those years ago, and sparked a chain of events that led to the formation of MRV and all of the great work that has come from it. So again, not wanting to necessarily dwell on sad events, but perhaps reflecting on the opportunities that have come from that really seismic event for her family and for many of her clinical carers that happened on this day. So thank you for reflecting on that. So we'll move over to Elizabeth shortly. So it's really my absolute pleasure to introduce Associate Professor Elizabeth Ng, who from the MCRI here in Melbourne, who really has done incredible work with understanding the basis of haematopoiesis, and really the ability for us as potential clinicians and researchers to springboard off of her achievements and discoveries for the development of new therapies with respect to haematopoietic stem cell reconstitution. Elizabeth will be presenting to us, and we look forward to her work and available for questions. So please stick around for that at the end of her presentation. You can read her qualifications there. We're very, very lucky to have her and her group here in Melbourne and as part of the broader Parkville research community. And I personally am looking forward to not only this presentation, but our future collaboration with her and Andrew's successful grant is, I think, about to be announced, if it's not, I haven't sprung it on you already. So with that, Elizabeth, thank you for joining us, and we look forward to your presentation.
Well, thank you very much, David. And can you all see my screen? Wake up.
Not quite yet.
As it's here. Let me try again. Okay, can you see anything yet? Actually,
No.
wait. Okay, how's that?
We can see you, for which we're fortunate, but I can't quite see your screen just yet. Oh, there's Andrew. Hi, Andrew.
It's working just fine. That's what I've done.
Here we go.
Yep.
Looks great. We can see that. Thank you.
Excellent. Well, thank you very much to you all for inviting me to speak with you today and tell you about the work that our team has done in generating hematopoietic stem cells from human pluripotent stem cells. Okay, let's see. So bone marrow transplantation, or hematopoietic stem cell transplantation, really was the first stem cell therapy to come into existence. And it was a key therapy for hematological diseases, and was established by the pioneering work of Donnell Thomas beginning in the late 1950s. And what he was able to show was that some patients who were treated were cured following chemotherapy and bone marrow transplantation. And these were really the first successful hematopoietic stem cell transplants. However, significant problems were noted that still persist today, such as leukemia relapse, which tells us that the therapy remains imperfect. And of course, as you all know, there is a significant amount of transplant-related mortality and morbidity, such as infections and graft versus host disease. So this tells us also that there is room for improvement and for the discovery of new and better therapies. So this story actually began a long time ago, and it's a result of a collaboration between Andrew, Ed, and myself. And you can see we've been working together since the dark ages. And in our long-term vision, what we envisaged was being able to produce engrafting hematopoietic stem cells from pluripotent stem cells that we could use for a number of different applications, including cell therapies, to devise new models of hematopoietic diseases, to study pathogenesis and improve treatments, but also to generate therapeutic blood lineages that would be of use either for screening purposes or for cellular therapies themselves. So why did this take such a long time? And it wasn't that it took us a long time, it also took the whole hematopoietic differentiation field a long time. And the reason for this is that there were an awful lot of ducks that had to align. Firstly, we can see that human embryonic stem cells were the first human pluripotent stem cell lines to hit the deck, as it were, in 1998 from James Thompson's laboratory. And then 2 years later from Martin Perra at Monash University. And at that time, this is when we moved over to Monash University to work with Martin and learn how to work with these cell lines. And then one of our first achievements was actually to learn to devise techniques to culture, passage, and cryopreserve the cells. And what this tells you is that we've spent a long time in the game. We've spent a long time learning how to grow, culture, maintain, and differentiate these cells. And this cell line here, the human embryonic stem cell line ENVI, was one of the first cell lines that we generated that had a constitutive GFP reporter and we're still using today. The other thing we realized when we were embarking on this work is that we needed to have robust, reproducible differentiation systems in order to differentiate these cells and to perform suitable analyses. And we needed to do this in an environment where there were no we needed to do this in an environment where we can actually control the differentiation. So one of the first things we did was we devised a first platform for differentiation of human embryonic stem cells, and this we called the spin embryoid body method. And what it was a very robust and reproducible way of differentiating the cells, and allowed us to assess the effect of individual growth factors on the differentiation of these cells. And we did this by also devising a differentiation medium that was neutral. So one of the problems we have in the differentiation field, which is we all know about, we don't talk about very much, is that most differentiation media are not neutral. So the differentiation conditions or media themselves can have an effect. And what we wanted to do was devise a system that was completely neutral, and so differentiation was controlled by the growth factors that you added into the medium. And it was also around this time that Shinya and Yamanaka's group published the discovery of induced pluripotent stem cells. And he used a cocktail of 4 factors, and a year later, another cocktail of 4 factors was also published by James Thomson's laboratory. So now for the first time, we knew that there were 4 genes in both the mouse and the human that could reprogram somatic cells to pluripotency. And what this meant was now matched pluripotent stem cells could be made from any individual. And this therefore revolutionized our ability to look at disease modeling and cell therapy. And it also circumvented the ethical concerns which the generation of human embryonic stem cells from human embryos had caused in society. So our approach to the problem of differentiating human pluripotent stem cells was to look at what we knew from embryology, from developing animal models, but also from developing humans. And what we aimed to do was to understand and also control differentiation by tagging one copy of a gene of interest with a fluorescent reporter. And this is what you can see here in the image. You can see that when in this case, we've tagged the RUNX1C gene. And what you can see here is when the cells are differentiated and the gene is expressed, the cells will fluoresce. And that allowed us to identify cells of interest when they appeared during differentiation, and it allowed us to control the differentiation. So one of the first cell lines that we made was a reporter for MIXL1. MIXL1 is expressed in the primitive streak of the developing embryo, and this allowed us to define culture conditions that would induce mesoderm from pluripotent stem cells. And this is particularly important because the hematopoietic system arises from the mesoderm. We were further able to then go on to generate reporter cell lines that had where we had tagged 2 genes of interest. In this case, we had tagged SOC17 with an M-cherry reporter and RUNX1C with a GFP reporter, so that we could identify both. And you can, if you look at my pointer, you can see the SOC17 cherry positive endothelial cells and the GFP expressing blood cells. And these reporter cell lines have been very valuable for many of the studies that we subsequently undertook. Now, because nothing happens in a vacuum, lessons from other model systems that we were also, us and other colleagues were working with, told us a lot about the mesodermal and endothelial origins of blood. So we knew from studies from looking at mouse models of differentiation where the first repopulating stem cells would arise, and some of the growth factor signaling requirements for this. Because what you have to remember is when we are looking at human developmental hematopoiesis, this area or this time of human development is virtually inaccessible. So they're we have at that time, very limited samples with which to compare our work. And also this, as I said, this time of human development is inaccessible. And also the embryo is very small. So what happens is the first mesoderm that's formed in the developing human migrates extraembryonically and forms the vasculature and the hematopoietic system of the yolk sac. And you can see that here, both in the diagram and in this transverse section here. And you can see there the extraembryonic blood islands. And then this wave of primitive hematopoiesis gives rise to erythroid and myeloid progenitors. And at the onset of circulation, these cells can actually enter the bloodstream. And this is a very early paper I've put down here, which describes these cells appearing in the human fetal liver during development. If we differentiate our human human pluripotent stem cells in this way, you can see that we can form SOX17 positive endothelium and RUNX1C positive blood. If we transplant these cells into a mouse, they're capable of homing to the bone marrow because they know that they're blood, but they do not engraft. And so the question we asked was, why? What's different? So what we did was compared our CD34 and RUNX hematopoietic cells that we were making in the dish that you can see over here, and we compared them to cord blood cells, which we know that no engraft. And what you can see here in the heat map on the right is cord blood cells express the genes of the HOX-A cluster. The HOX genes are switched on during anterior-posterior patterning in the developing human. And what you can see that this patterning is retained in the cord blood cells, but what you can see is our cells did not express these genes. So what we know is that during human development, definitive hematopoiesis begins intraembryonically. It begins in the AGM region of the human embryo, and we know that the first repopulating hematopoietic, and if you can, first hematopoietic cells are seen arising from the ventral wall of the dorsal aorta, which you can see in these immunohistochemistry images here, and they appear from about day 27. The first engraftable hematopoietic stem cells arise around day 32. So that tells us that most of the cells that are in the intra-aortic hematopoietic clusters are not hematopoietic stem cells. They're either pre-hematopoietic stem cells, but very rare cells do arise during this period of time. Following their birth in the AGM region, they then migrate to the fetal liver where they expand and then will eventually repopulate the bone marrow. So this is a process that we are trying to recapitulate in vitro. And as you can see from the embryology, it's quite a complicated process. During this time, work had been done on what other signaling pathways and factors influence mesoderm differentiation and regulate definitive hematopoiesis. And it was found that members of the Wnt family or and the TGF-beta family played significant roles in this. And what we were able to show in 2016 is that by using a combination of activin antagonist and Wnt agonist during mesoderm differentiation allowed the initiation of intraembryonic hematopoiesis in vitro. And what this allowed us to do was to develop an in vitro facsimile of dorsal aorta hematopoiesis, and you can see that here in the image where you can see SOX17 hemogenic endothelium giving rise to RUNX1C blood. So the key point about this is we knew that if you treated our differentiating embryoid bodies with this combination of molecules, it induced HOX-A gene expression within the developing mesoderm in culture, and it was this that went on to give rise to a facsimile of dorsal aorta hematopoiesis. And you can see here the SOX17 positive vessels giving rise to RUNX1C positive blood. We were also fortunate in being able to collaborate with Hanna Mikkola's laboratory, and at this time we were able to do some of the first comparisons of the cells that we were making in vitro with the early human embryo. What we were able to show is that hematopoiesis in the yolk sac does not express genes of the HOX-A cluster. To drive intra-embryonic hematopoiesis, you need to express genes of the HOX-A cluster. If we compare here on this top heat map, looking at human AGM, endothelium and stem and progenitor fractions, we could see that in our differentiating blood cells, we were expressing genes of the HOX-A cluster. We also knew from studies in the mouse that retinoid signaling is required to for HSC emergence in the AGM region. And when we looked at our differentiating cultures, you can see here again, looking at human AGM samples, when we compared our in vitro haematopoietic cells with the human AGM, that we could see that there was a deficiency in the correct retinoid signaling. So this, at the end of this series of work, it gave us the first hint that we possibly were on the right track to being able to generate repopulating hematopoietic stem cells from pluripotent stem cells, provided that we were able to pattern the cells correctly. We were also interested in order to carry this research forward, we had to also consider some of the further analyses that we were going to do. And so as part of the group, Ed Stanley's group developed the gap trap system, which enabled us to generate iPS cell lines that expressed constitutive colors. And these were extremely valuable because it allowed us to use these cells as workhorses to look at for engraftment studies in animals, because we were able to detect 1 in 200,000 cells within the bone marrow. And these are very, very early experiments that we did testing out these cell lines and whether or not we could detect them in the mouse. And you could see that, yes, you can see, and this is here using a cell that expresses the tomato gene. So then all this work culminated last year in this publication, which was the generation of long-term multi-lineage engrafting cells from human iPS cells. And actually, it required the coming together of a number of things. So the way we took all our differentiation systems and the knowledge we had of human development and put it together is as follows. We invented another version of the embryoid body system where we could differentiate human pluripotent stem cells in non-adherent swirling culture. So you can see here, instead of having one embryoid body per well, we can generate plates with hundreds of embryoid bodies. We used 2 workhorse cell lines, one that expressed the tomato gene and one that expressed a blue FP marker. This one happens to be the tomato gene, and what you can see during differentiation are the forming embryoid bodies. We drive them through mesoderm differentiation. We induce the HOX-A genes in the mesoderm and then drive them to endothelial and hematopoiesis. And what you can see here at day 7 of the differentiation are protrusions on the periphery of the swirling embryoid bodies. And these mark the in vitro equivalent of the hemogenic endothelium that exists in the dorsal aorta of the human embryo. And you can see with time, these protrusions grow and develop further, and they start to transition into blood, which is then released into the medium. And you can see at day 13 that the plates are full of blood. And this just happens to be a bright field image of one of these cultures. Up there labeled, you can see the dense body there, which is a swirling embryoid body. The structure on the side is the area of endothelial to hematopoietic transition. And you can see that the medium is actually full of blood cells that are released into suspension. So what do these cells look like in the dish? So the suspension cells that we have growing in the dish are CD45 and CD34 positive. So they're hematopoietic cells. They express CD90, c-KIT, and CD44, which are all markers of stem cells and progenitors. They do not express the lineage marker CD38, and they do not express CXCR4 or CD73, which are markers of artery and vein. If we look at the embryoid body component within the culture, you can see that there are two fractions. There's a CD90 positive stromal fraction and a CD90 CD34 positive endothelial fraction. We can max enrich for this fraction, as you can see here. And if we look at this fraction, it too can be subsetted into a haematopoietic component up here, which is CD45 positive, and an endothelial component down here, which is CD45 negative. The CD45 fraction is CD90 positive, CD44 positive, and again, CXCR4 negative, while the endothelial component is also CD90 positive, CD44 positive, and expresses CXCR4 and CD73. And what you can see here, that allows us to distinguish populations within that endothelial fraction that represent artery, vein, and hemogenic endothelium. So then we conducted a series of experiments where we looked at differentiating our tomato expressing cells or our blue FP expressing cells, and we differentiated them under a number of conditions. And what we were looking at was the effect of retinoid signaling during the differentiation. And then we performed single cell RNA-seq on these experiments. So this is quite a large data set. This work was done by Jackie Lee in our laboratory. And what you can see here is a UMAP of all the data. And within this UMAP, we can identify elements of stroma, elements of endothelial, arterial, venous, and hemogenic endothelium. We have an area of haematopoietic stem and progenitor cells, and we can also see that there are populations of haematopoietic cells that are showing markers of maturation. And mind you, remember that all these cells are CD90 positive and CD34 positive. So on the surface of it, they all look the same, but clearly their brains are not all the same. So the next thing that we wish to do with these is to benchmark the cells that we were producing in the dish with cells that are produced in the human embryo. And for this, we collaborated again with Hannah Mikkela's laboratory, who had mapped the emergence of human hematopoietic stem cells from the hemogenic endothelium stage in the embryo until birth. And what she had devised was a scorecard system to look at both emerging stem cells and maturation of stem and progenitor cells. And what she had identified here was a scorecard of these 6 genes that marked hematopoietic stem cells. So what Jackie did was he looked at the Carnegie stage 14 and 15 AGM samples from Hannah's data sets. And we know that this is the time in the human when the first hematopoietic stem cells are produced, and he reclustered them so we could look at this trajectory from artery to aorta, hemogenic endothelium, and to hematopoietic stem and progenitor cells. And he was able to show in this data set that the 6 signature genes were expressed, and that HLF and SPINK2 marked the most primitive of the stem cells. We took the cells that we had differentiated in vitro and did a similar analysis. So again, reclustered and looked at arterial, hemogenic endothelium, and hematopoietic stem and progenitor cells. And within these populations, we could also see the expression of the 6 signature HSC genes. Again, also a population that was SPINK2 and HLF positive. And this told us that our cells looked very similar to the first repopulating stem cells that are born in the human AGM in Carnegie stage 14 and 15 human embryos. Now, you remember that during this differentiation process, we were also looking at the effect of retinoid on the differentiation. And so what you can see here is, again, from this data set, we had samples that were not treated with retinoid, samples that had 1 pulse of retinoid, and samples that had a longer duration of retinoid. And what you can see marked in red here is the upregulation of genes that are of the retinoid pathway, and they are only upregulated if retinoid is included for the duration of the experiment. And what's important about this is we see these genes also expressed in the human embryo. So this told us that retinoid signaling was important for the differentiation of our hematopoietic stem cells, and in fact, a longer duration of retinoid was required to upregulate the genes in the signaling networks that they required. So the next part of this project was to functionally evaluate our IHSCs, and the gold standard functional evaluation is via transplantation. So we developed an algorithm where we would harvest our hematopoietic cells out of the suspensions. You can see we would immunophenotype them. So in this case, they were CD34, CD90 positive. We can cryopreserve them, and we did this purposefully, knowing that this is how clinical samples are treated. And then we can then transplant them via tail vein injections into NBSGW mice. And we would do the same thing with umbilical cord blood and, more recently, mobilized peripheral blood. We then analyzed the peripheral blood of the mice at 12 weeks and 16 weeks. And look for engraftment in the peripheral blood. We used a combination, particularly of CD43 and CD45, and these mark developing, maturing human cells in the peripheral blood. CD43 is not present in the mouse, so it's a human-specific marker. At 20 weeks, we then harvest the tissues from the mice and analyze the bone marrow, the spleen, and the thymus for engraftment. And we're also able to flush these tissues and cryopreserve the engrafted cells for further analysis. So what does this look like? What do these transplanted data look like? So what you can see at the top here in these first two FACS panels are images from bone marrow engraftment using the tomato cell line and the blue FP cell line. They are stained here with glycophorin and CD45, so glycophorin to mark the emerging erythroid lineage and CD45 to mark all the others. We were then able to go on and do similar experiments with two other independent cell lines which did not have a constitutive color, and you can see the patterns of their engraftment here. All four look very similar and compare also very well to our controls, which is cord blood mononuclear cells. What you can see here in this image is if you look in the bone marrow of a mouse that's engrafted, in this case with the tomato positive cells, you can see in there cells that are fluorescing red. So you can straight away see whether or not you've got engraftment in the bone marrow. If we look at the tissues of one of these engrafted mice, you can see here in the bone marrow that we have a population of cells that expresses tomato, and that they are glycophorin positive for the erythroid lineage, and the rest are CD45 positive. And within this 45 compartment, you can see we have CD19 B cells, CD3 T cells. We have myeloid cells here. And most importantly, in the bone marrow, you can see the presence of a CD38 negative, CD34 high stem cell compartment. When we looked in the spleen, again, you can see tomato positive cells in the spleen, and these are surface IgM positive. We can also see CD3 T cells. And in the thymus, we can see CD4 and CD8 positive lymphoid T cells. This panel, these couple of panels here on the end, show that in some of the samples, we would see a small population of CD19 cells. The thymus in these mice is very small and sometimes very, very hard to see or to dissect. And what we think here, where we've got some low thymic engraftment, is we've also taken some lymphoid cells, and hence we're picking up some lymphoid tissue, so we're picking up some CD19 cells. So if we look at I suppose the key findings of our paper, these scatter plots show the results of the bone marrow of mice engrafted with IHSCs generated from 4 independent cell lines. Here we see the tomato cell lines. You can see that there's a range of different colored dots. these dots represent either low-level myeloid or lower-level myeloid and lymphoid engraftment. But what I want you to look at are the red dots, which illustrate multi-lineage, high-level multi-lineage engraftment in the bone marrow and the spleen. And we saw this first with our workhorse cell lines that express the tomato or the blue FP. And we went on to be able to do similar experiments with two other independent iPS lines, which were uncolored. We also did a series of transplants with cord blood cells. We transplanted 39 mice with varying numbers of cord blood cells from 4 independent cords. They had roughly between 0.7 and 2.7% CD34 cells. And we did essentially a limit dilution and found that that 1 in 6,000, approximately 1 in 6,000 CD34 positive cord blood cells resulted in multi-lineage engraftment. And lower than that, you can see that there are other colored dots that appear in this scatter plot, which represent restricted lineage engraftment that can be seen from the cord blood transplants. And this tells us that that these oligo and unipotent progenitors that you are seeing in these transplants are actually a normal part of hematopoiesis. And they are similar to what we have seen during the transplantation of the iHSCs differentiated from our iPS cells. This is new unpublished data, and we performed a similar series of experiments with mobilized peripheral blood CD34 cells. And what you can see here, this is from two donors, is that roughly 1 in 113,000 CD34 positive cells would result in multi-lineage engraftment. But what you can also see is again, from these other colored dots, that restricted level engraftment in these transplants is also seen, and again demonstrates that it's a normal part of human hematopoiesis. We have then gone on to look at differentiating cell lines that have been made under GMP conditions, and that we have adapted to culture on recombinant human matrices, and also that we have differentiated using, if you like, a scale-up of the differentiation system in a flask culture. And what you can see here is one cell line, an LUMC cell line, where we've got a high-level multi-lineage engraftment, and also another cell line. This is an external cell line. So now we're starting to be able to work with cell lines that are brought into the institute. So roughly speaking, we have a total of 14 clonal cell lines from 6 independent iPS cell lines that have been grafted. And most importantly, the hematopoietic stem cells have been generated by 11 student and postdoc members of the blood group. And this graph here just shows you in total the analysis of all the multi-lineage engrafting mice that we have. You can see that generally, if we have multi-lineage engraftment, the contribution is very high, and these graphs here show the contribution of the individual lineages. And probably what is most important that comes out of here is the fact that each mouse is slightly different, even from the same population of cells. So each mouse acts as its own bioreactor. So how were we able to manage to get this to work? Well, there are several important points in the protocol. The first is how we generate the mesoderm and express the HOX-A genes. And you can see that mesoderm that's generated with a higher concentration of CHIA or Wnt signaling gave us multi-lineage engrafting cells. And furthermore, that if they required a pulse of retinoid, at least at one time point during the differentiation, and that also including it throughout the differentiation was also generated multi-lineage engrafting cells. The next thing we had to go and do was to look at the endothelium that we were generating in culture. And this, of course, is a consequence of VEGF signaling, which is required to specify artery during development. And what you can see here during our differentiation is under higher concentrations of VEGF, we could make very nice CD34 positive CXCR4 artery. But what we found, and this was again work we did in collaboration with Connie Bonifer and Alex Mayton, that if we, Hasindu in our lab, that if we removed if we left the VEGF in during the differentiation, you can see that the CD34 endothelial cells retained CXCR4 expression and retained an arterial phenotype. But if we removed it, the cells rapidly transitioned to the hemogenic endothelial quadrant that you can see down here, and continued to transition to there, and then gave rise to hematopoietic cells. And you can see here, if you look at a comparison of cultures where the VEGF was retained versus when it was removed, the multilineage engrafting cells emerge with higher frequency where the VEGF has been removed. So if we look into the bone marrow of the engrafted cells, what do we see? So again, this UMAP, we've only just started this work, so again, it's just beginning. What we can see are cells that mature in the bone marrow of the mice, and if we look into this section here, we can see hematopoietic stem and progenitor cells, and they have been grouped here, the hematopoietic stem and progenitor cells, in this kind of teal green color, but the most immature ones, which are HLF and SPINK2 positive, are here. We can see that this is the phenotype of the cells when they were engrafted. But what happens in the bone marrow? So here, the blue violins represent what the cells look like in vitro, and the green and red violins represent what they transition to after they've engrafted in the mice. So what you can see is that the expression of some of the stem cell genes is unchanged. So they were expressed in vitro, and they are retained in the bone marrow of the mice. Importantly, we can also see that the expression of other stem cell genes, which are lowly expressed in vitro, are actually upregulated as the cells engraft into the bone marrow and mature. So this would be the case for MLLT3, MECOM, HOXA9, and a number of others. Then we can also see that a number of other stem cell genes then are actually switched on after the stem cells engraft. In this case, ALD1A1 and PROM1 and genes in the HLA. So this tells us that the cells are maturing upon engraftment. And in fact, if we compare it to a scorecard of human embryo development, you can see that this is exactly what our cells are doing. So they are AGM stage when they are made in the dish, and with time of engraftment, the cells are able to mature. So what are our future work? Where are we going with all of this? So different members of the laboratory have now taken their genes of interest, and we have generated further numbers of reporter cell lines, so we can track the differentiation more finely and identify subpopulations within our CD34-90 populations which have that harbor engraftment potential. We'll also use them to look at lineage development. We have taken the plate embryoid body differentiation system we've got here and scaled it up into a FLAS system as a first example of scale up, and this is work by Maurizio Castro. And what we are trying to do is that we can show that we can transition our work from research into more of a bioreactor system. We're also looking at the ability of these cells to be useful workhorses for other systems, in this case to look at lentivirus and prime editing systems for gene therapies. This is some of the work done by Gulchan in our lab, who's looking at the lentivirus expression in the IHSCs, and Lorna McClemens' work with our colleagues at St. Vincent's. And what she's been doing here is taking IHSCs that express GFP and prime editing them so they express blue FP. We have other members of a lab, and this is an example of Jana Hagen's work where she's looking at differentiating neutrophils from the IHSCs, and what she's been able to do is devise a system and a protocol where she can generate functional neutrophils. We're also doing work where we look at the transplanted animals just to look at the distribution and location of the human cells when they engraft in the mice, and this is again work by Gulchan in our lab. And so I hope from just kind of giving you some of the vignettes of the things that we've been doing in the lab to be able to show you that we've created a differentiation platform for making CD34, CD90 hematopoietic cells with multilineage engraftment potential that we can use for cell therapy and disease modeling. However, it's been really, really important for us to be able to compare the cells that we generate in vitro with cells in the human embryo to show that we're tracking on the right developmental path. And this has allowed us to evolve the protocol and to look at, to gain insights into the mechanisms that drive HSC emergence from the hemogenic endothelium. And that's allowed us over time to improve the protocol robustness and efficiency. We've been able to show that cord blood, our IHSCs, cord blood and mobilized peripheral blood display similar engraftment outcomes, and that the IHSCs can mature in the stem cell compartment. We've been able to show that we've been developing reporter cell lines to help us refine and understand this process better, and that we're transitioning the work into systems where it allow for the scale up of our blood differentiations for future clinical transition. And finally, this is work that we have done in collaboration with our partners that are here on the slide, because our vision for the next stage of the process is to develop what we call a P2P autologous cell therapy pipeline, which is a patient to patient, in which we can actually take patient cells, reprogram them, and drive them straight into a differentiation pipeline and generate them a CD34 IHSC product for autologous cell therapy. And as David mentioned before, this is again part of a grant which is yet to be announced, that we hope that the clinical translation of the results of this project will change the landscape around hematopoietic stem and cell transplant therapy by ushering an era of personalized therapy for patients with leukemia and bone marrow failure. And in order to do that, we need to establish a dialogue with patients and families and advocacy organizations, not only just to bring them along on the journey, but to consult with their wishes and concerns and the scientific directions of this work, and also to devise the most suitable product that will be could be used in a clinical situation. And to this end, we've formed strategic partnerships with Maddie Riewoldt's Vision, the Leukemia Foundation, and the Arrow, and the Bone Marrow Transplant Foundation. And with that, I would like to thank the people who did the work and the differentiations, who are all here, and the important senior people, and the members of the blood and immune development groups as a whole. Thank you very much.
Well, thank you very much, Elizabeth. That was absolutely fantastic. And thank you also for leaving us a little bit of time for questions.
I'm 3 minutes long.
Oh, darn it. Well, I took a long time to do the introduction, so we'll call it even. So I don't think we've got capacity for live questions, but Tracy has popped one in the Q&A window, which I'm not sure if you can read for yourself, but let me read it out for everyone else's.
Yes.
Yeah.
Yes.
Are there any, sorry, you read it and answer it.
Are there any safety concerns associated with the introduction of the Yamanaka factors, or are they all downregulated during the differentiation process? In the process of making the iPS cells, those vectors are not retained. So once the iPS cells are in existence, after several passages, the cells are normal. So then the next part of the question is, are they all downregulated during the differentiation process? So during the differentiation process, we can, and again, we've got data which we haven't shown, is we can show that in actual fact, the cells traverse normal differentiation to mesoderm and endothelium. So they express what we would expect to be a normal gene expression profile.
And so the answer to that is that there should be no safety concerns.
No.
Thank you.
in the reprogramming process, one of the other things that we're actually testing at the minute is actually using other forms of the reprogramming process, such as mRNA, which doesn't hang around either. But mind you, the reprogramming process that we use at the minute, even with the Sendai virus, is scarless. So there's no retention of any of those reprogramming genes.
Very good. Any other questions? If there are questions, pop them in the question and answer window. I've got a question, Elizabeth. So you used, Dan North's asked a question, we'll get to him in a sec. You used the term through your presentation, robustness of generation and robustness of systems, all of which have been fantastically demonstrated. What about the robustness of the cells themselves, in the sense of once they've engrafted, do they continue to provide a robust source of self-renewal? And if I can just illustrate that with one little bit further, we would wish as clinicians that transplantation is the end of our patients' clinical needs, but sometimes it's not. Sometimes there are ongoing insults. They may be chemo or immunotherapy. They may be immune-driven, particularly in the allogeneic setting. Most commonly, they may be inflammatory in the setting of infection, all of which pose potential insults to hematopoiesis. And have those been tested in your induced model?
So we haven't done what I would call insult testing yet, although possibly the nearest plan we've got to do that is to test. Looking at the problem, I suppose, from another point of view, is to test, for instance, the functionality of the neutrophils in infection mouse models. So that will tell us whether or not neutrophils that are derived from iHSCs can actually tolerate an infected environment, as it were, and perform. So we haven't done those experiments per se. You asked also, I think, about self-renewal, and the best, I think, self-renewal experiments are secondary engraftment, which we have just begun to do but haven't got very far yet. But the point I would make is that possibly some of your questions can't be answered in a mouse model, because we know that the mouse is a poor host for human cells. We have a collaboration with the JAX lab, who have a number of different humanized mouse models that we are going to try whether or not we can look at making different sorts of humanized mice under these conditions and see how our iHSCs perform.
Excellent, thank you. Sort of two linked questions in the chat, in the question and answer, one from Daniel North and one from Claire, about one of the potential recipients, beneficial recipients of this work, are patients with inherited or germline bone marrow failure syndromes. Do you foresee, or have you actually been able to undertake any of the induced pluripotent cell work in the presence of germline abnormalities?
So the work that we've begun so far is we also have a collaboration with the SDS Alliance. And so what we have started doing is we have started to look at that particular problem, looking at patient-derived iPS cell lines. Now, there's probably two approaches, none of which we know the answer yet. And I'm not a clinician, so probably some of the things I'm going to say are going to be quite naive. However, one of the ways that are looking at this from a science point of view is, first of all, there's the possibility in the future, but not at the minute, I should think, that we can gene edit for germline mutations that we know about. So we can make gene-corrected iHSCs that are autologous. That's the first point. And we've done this with an SDS model for shock and diamond syndrome. And what we are looking at is whether or not the gene-corrected cells can outcompete the uncorrected cells. The other thing that we can possibly do, depending on the genetic mutation, is that we can try deriving iPS cells from tissues of the patient that don't express. So for instance, with one of the problems we're trying to deal with bone marrow failure patients is they have very few CD34 cells. And so we're looking at a combination of generating iPS cells from mesenchymal cells or fibroblasts or other cells. And I think that sort of answers Claire's question as well. Is the
yeah, I think they're
if you have an identified germline defect, that can be gene corrected.
very good. Any other last minute questions? Pop them in the Q&A if you think of them. Here's Steve. So just asking about your scale up. You can have a look at that. Did you use ex vivo haematopoietic stem cell expansion factors, or was it really just a volumetric increase from your animal experimentation?
So essentially, we do the whole differentiation rather than in plates. We do it in flasks. So essentially, we make 300 million cells or whatever per flask. So it's not actually the same as taking. I know people have looked at taking cord blood CD34 cells and trying to expand them in culture and still retain repopulating ability. We know that ability drops with time in culture. So instead, what we're doing is we're trying to expand the system such that we can generate patient doses of these cells to cryopreserve. We are testing the first differentiation experiments that I talked about that are in the paper. We did not use UM171. We are aware of UM171 and its use in expanding cord blood HSCs, and we're actually at the minute testing it on our iHSCs. So we use a very, very limited cocktail of differentiation and factors to produce and maintain the iHSCs.
Excellent. Thank you very much, Elizabeth. I've just got a couple of things I need to deal with, but thank you so much for sharing your expertise and your time with us today, Elizabeth, and on behalf of all of your very many industrious and incredibly intelligent scientific colleagues and students, and the 465 plus 45 mice I think that you had on the last slide.
Exactly.
Thank you to them. Just before we round off and before people leave, I do just want to thank our sponsors, and so Novartis has helped us put together this national series of symposia, so we're really really indebted to them. But equally, if not more importantly, MRV is looking at always new ways to engage the community, patients and their families, and the broader sort of scientific membership. And so MRV, as part of its 10th anniversary, as you've heard about me talking about at the beginning of the meeting, there's the National Bone Marrow Failure Syndromes Awareness Week. This is an initiative launched by MRV, and it starts on the 3rd of March, which is just next week. It's March already, people. This is a week-long campaign that the team at MRV, and particularly spearheaded by CEO Amy, have put together to raise the awareness regarding bone marrow failure syndromes. More awareness means greater equity of access to the patients, and hopefully greater support from the community to allow ongoing research opportunities, some of which we've heard about today. It really does highlight maybe Riewoldt's Vision's leadership in making that research opportunities available to researchers young and old. And that this is in partnership with Stem Cell Donors Australia and Lifeblood. And so you may see some promotional advertising material around the place, as well as a social media program. If you want to participate, your expertise, time or knowledge in various ways, you can do so through social media channels, or indeed connect with J.A. Sherry or Amy directly, or Zoe Austin, who's our new marketing and communications manager. So let's get out there together collectively and promote the understanding, the knowledge of bone marrow failure syndromes. That's the way to bring much needed funds into the field and to deliver meaningful outcomes through research, just as you've heard today. So let's stay engaged with that. The other thing, just a quick plug, is that on the 23rd to the 25th of May, here in the VCCC building, will be our 4th National Symposium on Bone Marrow Failure Syndromes. So get early registration and get along and participate in what is always an excellent meeting with international and national speakers. So lots happening in MRV in its 10th anniversary, and no better time to recognise that than today, the 10th anniversary of Maddie's passing. We are really keen on involving these symposia, all of the symposia and all of the webinars that we offer and all of the engagement. So if you do get a questionnaire for participating today, please fill it out. The team in the office are definitely interested in your responses to help us redesign and design and continue to evolve our offerings. So with that, thank you particularly to Elizabeth and her team. Excellent work, really super, and super exciting about what the next couple of years may hold. And thanks to all of you for coming along and the team at MRV for making it all possible. Okay, night everyone, thanks very much.